Hello and welcome everyone to the channel Biology Explained. Today I am back with another video in which we are going to discuss the question paper of CPET Zoology Entrance Examination. And this question paper is of the year 2024. So this is the part 2 of this video, and part 1 I have already uploaded it. In part 1 we have already discussed up to question number 39. Here we will begin from question number 40. So let's begin. So question number 40 is, during splicing, which of the following site is not recognized by the spliceosome? So as you know, splicing is a post-translational modification in which the introns are removed and exons are joined of the mRNA. Okay, so splicing, the mRNA which is undergoing splicing is having three sites: a 5' splice site, a 3' splice site, and a branching site. As you can see in the diagram, here is the 5' splice site, here is the 3' splice site, and here this point is the branching point. So three sites are there. So the question is asking not recognized. Not recognized, which is not recognized? 5' splice site is present, branching point is also present, and 3' splice site is also present. Therefore, the answer for this will be option number A, that is the G-rich site. Additional information you can remember: the 5' splice site is rich in GU sequence, the 3' splice site is rich in AG sequence, and of course you can remember the sequence of the snRNPs, because this is frequently asked in the questions, like after which snRNPs are coming and how the spliceosome complex is formed. Okay, next, question number 41. Question number 41 is, the studies that led to the discovery of post-transcriptional gene silencing phenomenon were performed on sunflower, petunia, maize, parsley. So the answer for this will be option B, that is petunia. Here for your convenience I have kept this diagram of petunia also, you can see it. The next question, that is the question number 42. Question number 42 is asking about a lethal mutation. Okay, the lethal mutation that is seen in yellow mice is due to which mutation? Remember, this mutation is lethal in which condition? In homozygous condition. In homozygous, if a lethal mutation is present in homozygous condition, the mice will die in the embryonic stage itself. But in heterozygous condition, a yellow coat color will be expressed, and this yellow coat color is due to mutation in which gene? In the Agouti gene. Agouti gene, and this is a type of insertional mutation. Okay, this is what you need to remember. So the answer for this will be option A, that is insertion. Next question, question number 43. Question number 43 is, if a gamete has 16 chromosomes, what will be the number of chromatids before anaphase 1? So this is a concept from meiosis that you must know, that meiosis as such, when we discuss meiosis and mitosis, which one is reductional? Of course, meiosis is reductional type of division and mitosis is equational. But in meiosis consists of two phases, that is meiosis 1 and meiosis 2. So among meiosis 1 and meiosis 2, which one is the reductional event? Remember, meiosis 1 is the reductional event and meiosis 2 is equational in nature. As such, between mitosis and mitosis is equational and meiosis is reductional, and in meiosis particularly which phase is reductional? Meiosis 1 is reductional in nature. Okay. Let's see. The question is saying that there are 16 chromosomes. Gamete has 16 chromosomes. Gamete means always gametes are haploid in nature. So the diploid level will be how much? Diploid will be 32. Okay. So what will be the number of chromatids before anaphase 1? Okay, let me explain this with the example of with the help of this diagram. Here you see the gamete, it is saying that the gamete has 16 chromosomes. Okay, gamete has 16 chromosomes, and we know that the parent cell will have how many chromosomes? 32 set of chromosomes. Okay, it will have 32 chromosomes. Then it is asking that what will be the number of chromatids in before anaphase 1. Before anaphase 1 means the chromosomes are not split. Okay, there is a common confusion, let me clear that also, that which one is one chromosome. This is the structure of chromosome, single chromosome. The single chromosomes looks like this. Then what is this? What is this X-shaped structure? This is also a single chromosome. Both are one chromosome only. Then why it is X-shaped and why this is rod-shaped? Because during cell division, what happened? DNA number duplicates. As you know, as you have studied cell cycle, then you must know that amount of DNA duplicates. The number of chromosomes remains same. This is one chromosome. This is also one chromosome. Here you can see here rod-shaped structure there. This is single chromosome. Here X-shaped structure also represents a single chromosome. So what happens during because there is DNA replication, so there is addition of these additional chromatids, means sister chromatids develop during cell divisions. Okay, before cell division there are no sister chromatids in the chromosome. Okay, this is a key concept that you must know. So here the question is saying what is the number of chromatids before anaphase 1? Before anaphase 1, the chromatids are not separate, means the chromosome has not split into two. Okay, the chromosome is not splitting. So if the chromosome is not splitting and there are 32 number of chromosomes, what will be the number of chromatids? 32 into 2. That is how many? 64. So the number of sister chromatids will be how many? 64. So option D will be the right answer. Clear? Next, question number 44. Question number 44 is asking the direct repeats in insertion elements are present. Direct repeats in insertion elements are present within the transposons, upstream of the inverted repeats, within the inverted repeats, and downstream the inverted repeats. Okay, remember a transposon elements is made up of, if I'll show the diagram of a transposon, here you can see the transposon element is made up of sequence called inverted repeats are present. Here this is the structure of a transposon. Here you can see inverted repeats are there, and here is the transposase gene. Here is the transposase gene, that is the transposons, and it is present when it is inserted into the target site. It creates some direct repeats. Here you can see when this gene is getting inserted into target sites, here direct repeats are created, and inverted repeats is something that is present along with the transposons itself. Okay, the question now is asking direct repeats in insertion elements are present where? Within the transposons, upstream, or within the inverted repeats, or downstream of the inverted repeats. So the best answer for this will be, if I draw the sequence of transposons will be like this. This is the transposon gene. Okay, here both sides what are flanked? Inverted sequence are flanked. Okay, this is the IS sequence, and ultimately when the gene is inserted into the target site, then we get direct repeats on both these sides. These are direct repeats. Okay, direct repeats are present. It is asking direct repeat in insertion elements are present where? Okay, you can see it is flanking both the sides. So the best answer will be not upstream. It is not only present in upstream, it is present in downstream also. Only upstream will be wrong, only downstream will be wrong. Within the inverted repeat, it is not possible. Inverted repeat is present beside the direct repeats, isn't it? So this option will be wrong, and within the transposons, this option is the most accurate. So with the elimination method, you can find the answers of this question. Okay, so the answer of question number 44 is A, that is the within the transposons. Next, question number 45. Question number 45 is, in chick development, the cell movement during gastrulation is called what? So you know the morphogenic movements are present, different type of movements are present. Basically in chick we see two types of layer are formed. One is epiblast, another is hypoblast. So these epiblast and hypoblast are formed due to a type of cell movement. The question is asking cell movement. It is not asking the result. Be careful. Most of the student will mark it as delamination, but delamination will be wrong. The right answer for this will be option C, that is ingression. Let me show you in more detail. Okay, here you can see this is the two layers which are formed. Okay, the upper layer is epiblast and the lower layer is hypoblast. So hypoblast layer, this epiblast and hypoblast layer, how it is formed? Because cells from the upper layer are migrating to the lower layers. You can see Okay, when this migration is happening due to a cell movement, and this cell movement is called your ingression. So the right answer will be option C, that is ingression. I have kept a diagram of this morphogenic movement. You can take a screenshot of this for your convenience. Okay, and remember the examples also. So next question is question number 46. The question number 46 is asking, for an unborn child, which of these procedures has least risk? Okay. So the options are A, alpha fetoprotein sampling, B, amniocentesis, C, chorionic villi sampling, and D, all have the same risk level. Let me tell you what are these. Alpha fetoprotein sampling may be a new term to you. Let me explain each and every options. Okay. Option B is amniocentesis. Amniocentesis, you know, from the mother's womb. Okay, mother's womb, what we do, the fetus is present in the mother's womb. So what we do, there is amniotic fluid in which the fetus floats. So we extract this amniotic fluid and we examine it, and this happens where? In amniocentesis. Okay, so there is physical extraction over here, and this projects 0.01% risk to the fetus always. Next is chorionic villi sampling. Chorionic villi are what, you know, the fetus is made up of two layers. Outer layer is trophoblast and the inner layer is inner cell mass. Okay, outer layer is what? Trophoblast layer, and the inner layer is called the inner cell mass. Okay, so trophoblast layer itself, they form what? They form the chorionic villi. Okay, so chorionic villi sampling, what we are doing, we are taking a small sample. From where? We're taking a small sample from this trophoblast layer and we are examining it. So this is also a physical process, and this may project some sort of danger, some sort of risk to the fetus. Okay, so amniocentesis and fetal chorionic villi sampling, both are risky. And third, the first option, which is your alpha fetoprotein, I'm telling you what happens when a female is present. There is a protein which is called alpha fetoprotein, or in short we call it AFP. Okay, AFP, alpha fetoprotein, that is secreted from the liver of the pregnant female. Okay, this level of alpha fetoprotein increases during the pregnancy. So what we can do, this is secreted in the pregnant women, and this level is present in the blood itself, female blood. So without affecting anything in the womb, we are not doing anything in the womb, we are directly taking a blood sample from the mother's body and we are examining the AFP content. Okay, what is the AFP content in the mother's body? We are examining it and we get to know about the disease condition of the fetus. Okay, which disease are primarily found in this, like it can help to detect liver cancer of the fetus, any sort of chromosomal abnormalities can be detected, and also spine tube defects can be detected, particularly a condition called spina bifida. Okay, spina bifida. Okay, this can be detected with this AFP test. So there are numerous tests available to detect the any problems with the unborn child. The most appropriate is the AFP test because this is least invasive. In amniocentesis, we are drawing blood from the amniotic fluid. In chorionic villi sampling, we are drawing a sample of chorion. But in AFP, we are not at all touching the fetus. We are just taking a blood. We are just doing a blood test. So the right answer for this will be AFP. Next, which type of placenta in ruminant ungulates is? Okay, there are different type of placentation. The different answer for this will be option B, that is syndesmochorial placentation. I am telling a brief explanation. How can you remember the types of different placentation, because there are different types of placentation based on the degree of the attachment. Always remember, there are 3 layers from the fetal side and 3 layers from the mother's side. Okay, as you can see in this diagram, this is the degree of placental union. There are 3 layers from the fetal side, as I said, and 3 layers from the mother's side. Okay, from mother's side, I'm telling you one by one. Here you can see the mother layer. First layer is the uterine epithelium is there. Then after that, the uterine tissue, and the uterine tissue, as you know, it is called what? Endometrium layer. And then comes the endothelium means the blood vessel of the mother. Okay, 3 layers: epithelium, then the endometrium, then endothelial layer. Endothelial means the blood vessel layer. Okay, 3 layers from the mother side, and the fetal side also 3 layers are there. Fetal word, first layer will be, fetals will be having a chorionic layer, isn't it? They will have a chorionic layer, then chorionic layer, then there will be tissue of the fetus, and there will be blood of the fetus, blood or the endothelium layer of the fetus. So these 3 layers, 6 layers in total, 3 from the mother side, 3 from the fetal side. I am repeating it again. 3 layers from the mother sides are the mother's uterine epithelium, okay, epithelial layer, then the endometrium tissue, and the endothecium. Okay, endothecium. Endothecium means the blood vessels of the mother. Okay, 3 layers from the mother side. Fetal side again, the chorionic layer. Chorionic layer is there. Okay, then the tissue of the fetus, and then the blood vessels of the fetus. Clear? So based on the degree of attachment, how much they are attached, they are a different type of placentation. A simple way of remembering is, first example I am giving you. First example is the pig and horses. Pig and horse have this epitheliochorial layer. In this first type of placentation, you can see there is only contact with the epithelium of the fetus, epithelium of the mother's uterus and the chorion layer, isn't it? Epithelium and chorion layer. That is why this is called epitheliochorial. And the second option, that is a B. B, you can see this is syndesmochorial, which is found in the ruminants. Okay, what is connected here? Here epithelium of the mother is absent, isn't it? What is present? It is the endometrium layer of the mother is present, that is attached with the chorion layer of the fetus, isn't it? That is why this is called syndesmo. Syn means fused. Syndesmochorial, and this is found in the ruminants. Okay, next, in case of humans I am showing you, that is D. You can see this is hemochorial. Hemochorial means there is a tight binding with the blood. There is tight binding between the maternal tissue and the fetal tissue. Here you can see there is no epithelium of the mother is present. Okay, only the endometrium layer of the mother is present, and that too is fused up to an extent that blood vessel is also fused, isn't it? So blood vessel, you cannot see a blood vessel over here. Here the blood vessels are there. Here you cannot see the blood vessel because the blood vessels are fused with the layers of the fetus itself. So this is hemochorial, means the tissue, the fetus is directly bathing in the blood itself. Okay, hemochorial. The chorion layer is in contact with the blood of the mother. That is why it is hemochorial. Likewise, you can remember. Okay, these are the 5 types of placentation based on the degree. I hope it is clear. If you are not clear, then rewind it again, you will understand. This is very simple. 3 layers from the mother side, 3 layers from the fetal side. Okay, and based on which layer they are getting attached, that is, and accordingly the names are there. How much the degree of attachment is there. Okay, next. So next question number 48. Miocene is an epoch between, so this is purely factual question. For this you need to remember the entire geological time scale. So let's see, Miocene is an epoch between which 2 phases? It is Pliocene and Oligocene. So which option will be correct? Okay, so the option A, that is Oligocene and Pliocene, will be correct. For you can remember the additional ages, like for age of amphibians it is Permian period. Okay, then your age of fishes, that is Devonian period. So these are the some sort of questions which are frequently asked. Beside that, try to remember the sequence. Next question number 49. Question number 49 is Pithecanthropus erectus. The first fossil of Australopithecus was discovered by Du Bois from, okay, Pithecanthropus erectus is another name of Homo erectus, and as you know, Homo erectus was discovered in Java, Indonesia. So the right answer will be option B, that is Java, Indonesia. Additional information that this Homo erectus is also called Java man or Peking man. Okay, this is the same thing. Next, question number 50. Okay, so this is the question from evolution. The gradual evolution of species that continues to exist as an interbreeding population is known as, if you have studied evolution, you must have come across 2 terms, anagenesis and cladogenesis. So what are these Anagenesis is what? Anagenesis is direct gradual evolution is called anagenesis, whereas cladogenesis is descent by branching. Okay, here you can see in case of anagenesis what happens, species 1 gradually evolves into species 2, whereas in case of cladogenesis, that is branching. So option A will be the right, because this question is asking about gradual evolution. Okay, gradual evolution, that means anagenesis is right. Next, question number 51. The great dying occurred during the great dying. What is great dying basically? Great dying means there was massive destruction, massive extension of biodiversity during this period. So the great dying is which period is recognized as the great dying? Which mass extension is recognized as the great dying? It is option C, that is the Permian and Triassic mass extension. There were 5 mass extension which you need to remember. That is, first one is the Ordovician Silurian mass extension, then followed by Devonian mass extension, then the Permian-Triassic mass extension. That is a third mass extension which is also called the Great Dying, and then fourth is Triassic mass extension, and the last is Cretaceous mass extension. This is also there in your syllabus. KT extension, you know, na? Cretaceous Tertiary mass extension. Okay, so the right answer for this will be Great Dying was in Permian-Triassic mass extension. So option C is the right answer. Next question. Question number 52. Okay, domesticated rabbits were released into isolation in an island for generation of the total population. 9% were black due to recessive allele. What could be the heterozygous percentage of this gene? Okay, this is a simple question from Hardy-Weinberg equilibrium. You need to remember two formula. One is P plus Q is equal to 1. Second one is P square plus 2PQ plus Q square is equal to 1. Okay, so here what is given? Here the recessive 9% is given as the recessive allele. When recessive allele they are speaking, then we have to take either P square or Q square. Let's consider Q square to be recessive. Okay, so what is the value of Q square given? Q square is 9%. Okay, it is 9%. 9% means I can write it as 9 by 100. That is 0.09. Okay, so Q square, the value of Q square is 0.09. If the value of Q square is 0.09, then what will be the value of Q? Q will be root over of 0.09. That will become how much? That will become 0.3. Okay, that will become 0.3. So now we have the value of Q. If we have the value of Q, we can find out the value of P now. P is equal to 1 minus Q, that is 1 minus 0.3, that becomes 0.7. So what the question is asking? Heterozygous percentage. Heterozygous means we need to find the value of 2PQ. So simple, 2PQ will be now equivalent to 2 into 0.7 into 0.3. What it will become? It will become 0.42. Okay, 0.42. 0.42 we can write it as 42%. So the right answer for this will be, what could be the heterozygous percentage? It is asking heterozygous percentage. Answer C, that is 42 will be the right answer. Next question. An example of associative learning would be, okay, what is associative learning? Let me tell you. Association means when associative learning occurs when an organism learns a relationship between two stimuli or stimulus and a response. Means there will be a stimulus and a response. Okay, if there is a relationship, if an organism is learning through stimulus and response, it will be called associative learning. Okay, two main experiments were performed that you need to remember. First one is the classical conditioning and the second one is the operant conditioning. What is classical conditioning? Classical conditioning, you must remember the dog and the bell experiment which was performed by the Pavlov. In this, the stimulus what stimulus was the bell sound, and the response was the dog started salivating upon hearing the sound of the bell. So stimulus and response is there. Okay, so classical conditioning is a case of associative learning. As you can see, what are the options? First is classical conditioning. So this one is right. Let's see at the other options also. The second option is operant conditioning. What is operant conditioning? Operant conditioning experiment, you remember the Skinner box experiment, okay, in which was performed by B.F. Skinner, in which what he did, he put a mice and he was putting two buttons. Upon pressing a particular button, food was coming out. So what happened? Gradually the mice learned that upon pressing a particular button, so button is a stimulus and he is getting a output, that is the food. So he is relating the stimulus and response. Okay, so operant conditioning is also correct in this. Pavlovian conditioning is same as the classical conditioning, because classical conditioning experiment was performed by Pavlov. So option C is also correct. So we have a fourth option, that is all of these. So the answer for this one, question number 53 will be D, that is all of these. Next question, Skinner box. This is the same question I have just explained. Skinner box is used for the experiments in case of operant conditioning. Option B. Next question, question number 55. Question number 55 is, body's primary circadian pacemaker is pineal gland? No. It is hippocampus of the brain? No. Amygdala? No. It is the suprachiasmatic nucleus. Option D is the right answer. Okay, hippocampus has memory functions, memory formation functions, then amygdala is responsible emotional processing. Pineal gland secretes melatonin. It does not controls the biological clock directly. So most of the students will do mistake here that they will mark pineal gland. But remember, it is not directly controlling. As you can see, the suprachiasmatic nucleus is located in the hypothalamus and act as the master of the biological clock. It receives light signal from the retina and synchronizes the body rhythms with day and night cycle. As you can see, it receives the light signals from the retina and accordingly it synchronizes the body cycle. Okay, that is why we are advised not to have late night use of mobile phones, that will affect our biological clock. So next, question number 56. Cos site of a cosmid, options are, consist of 12 bases, this is right, helps the whole genome in circulation and ligation, this is also right. So contains cleavage site, no, this is wrong. So option C will be the right answer, that is both A and B are correct. Next, question number 57. In 1990, the first gene therapy was given to treat. Okay, this is a direct question and this is a repeated question also. This was the first gene therapy that was given to treat was ADA deficiency. ADA deficiency. Here in the option you can see option D, adenosine deaminase deficiency. So option D will be the right answer. Next, question number 58. Question number 58 is, which hormone is administered to a person suffering from a marked fall in blood pressure? So which hormone is given when blood pressure is decreasing? So the right answer for this will be option B, that is adrenaline. Adrenaline, as you know, adrenaline is also known as epinephrine. It is released during emergency as a stress response, and this is responsible. Adrenaline hormone is responsible for increase in heart rate, then also increase vasoconstriction, rise in blood pressure. So this is helpful for this purpose. So option B will be the right answer. Who submitted the first patent for CRISPR-Cas9 technology in 2012? So CRISPR-Cas9 is as such not there in your syllabus. This is a part of general knowledge. If you know, in 2020 actually 2 females were awarded Nobel Prize for this CRISPR-Cas9 technology, and this was a Nobel Prize in chemistry also. So this Nobel Prize was awarded to Jennifer A. Doudna and Emmanuel Carpentier. So option B will be the right answer. Next, question number 60. Tetany is caused by, options are A, hyperparathyroidism, B, hypoparathyroidism, C, hyperthyroidism, D. Deficiency of mineralocorticoid. For questions like this, you must remember the actions of the glands, the actions of the endocrine gland, which is there in your syllabus. As you know, parathyroid gland is responsible for the regulation of the calcium. Okay, and if there is a lack of calcium, it can cause tetanus. Okay, tetany condition. So this is how it affects the body of the affected individuals. So right answer for this will be hypoparathyroidism. Question number 61. What method can be used for carrying out in vivo fertilization of a sterile but fertile female? Okay, this question is quite tricky. What is saying? The female is sterile, but again it is saying fertile. So two words are there. When we will say a female is sterile, when she is not able to produce the ovums or the eggs. When the eggs or ovums are not produced, she is sterile. But the question is saying that she is sterile, but she is fertile. Fertile means anyways the egg is being produced. She is producing the egg. But why she is sterile then? She is sterile maybe because she is having some other issues with the implantation, or she must be having some other issue with the number of sperms the male counterpart is producing. So there is a number of reasons because of which a female is said as she is sterile. So here in this question, the female is producing eggs successfully. Okay, the female is producing eggs successfully, but she is not able to conceive. So which type of method can we adopt for this case? We can use ZIFT, that is zygote intrafertile fallopian transfer. No, ZIFT is not required because she is producing the egg successfully. Next one is gamete intrafallopian transfer. Yes, this can happen. Why? If the male sperm is transferred here to the fallopian tube, this egg is successfully produced, it can fuse with the egg in the fallopian tube itself. So GIFT will be the right answer. That is option B. Next question number 62. Which radioactive isotope is commonly used in RIA? What is RIA? Basically, RIA, as you know, it is the radioimmunoassay, and this is used to detect very small amounts of hormones or antigens. So particularly for this RIA, we use hydrogen, particularly hydrogen 3 isotope, which is also called tritium. Okay, tritium. As you know, we have 3 isotopes of hydrogen, deuterium and tritium. So hydrogen 3, that is tritium, is often used for RIA. And other commonly used isotopes also include iodine 125. Okay, we also use iodine 125 for this RIA. Okay, next question number 63. MHC class 1 molecules are primarily involved in. Okay, for those who don't know, let me tell you, there are 2 classes of MHC. MHC stands for Major Histocompatibility Complex. Okay, it is Major Histocompatibility Complex. And what are MHC? MHC are some receptor molecules. Okay, these are receptor molecules. So 2 classes of MHC are there. One is MHC1. Another class of receptor molecule is called MHC2. Okay, this is called MHC2. So what happens? MHC1 molecule is present in all nucleated cells. It is present in all nucleated cells. And MHC2 molecules are only present in some specific immune cells. Okay, specific immune cells like dendritic cells, macrophages, they all have this MHC2 molecules. Others have MHC1 molecules. So what this question is asking, what is the function of MHC1 molecules? So recognition of glycolipids antigens. MHC molecules, MHC1 or MHC2, they do not recognize glycolipids, they recognize peptide antigens. So this is wrong. Option B, resistance to fungi. Resistance to fungi will be wrong because fungi is an exogenic agent. Okay, exogenic agents means that is acting on the surface. Okay, that is acting on the surface. So if something is acting on the surface, these receptor molecules are not needed. MHC1 and MHC2 molecules, what they do is they show the intracellular antigens. MHC1 particularly, MHC1 function is to show the intracellular antigen. Suppose there is some problem in this cell. Suppose there is some infection by any virus. Okay, some virus has infected this cell. So what will happen? An intracellular antigen will be represented here by MHC1, and this will be recognized by your cytotoxic cells. Cytotoxic cells, particularly cytotoxic killer cells. Okay, that is the CD8 cell. CD8 cell will recognize it, and it will ultimately kill it. So virus infected cells shows this MHC1 complex. Okay, that is present in MHC1 complex. So the right answer for this will be option C, that is resistance of viruses. Okay, it helps in resistance of viruses. Activation of neutrophils, this is again wrong. For activation of neutrophils, we have cytokines and other molecules. MHC complexes, that is major histocompatibility complex, are not involved in this. Next question, question number 64. An example of live attenuated bacterial vaccine is, okay, what is a live attenuated bacterial vaccine? Live attenuated vaccine contains weakened forms of a pathogen. Suppose a virus or a pathogen is there. Okay, suppose any virus is there or any pathogen is there. Its weakened form is present in live attenuated vaccines. Okay, so the right answer for this will be B, typhoid. Remember one simple hack, because live attenuated viruses injecting is a very dangerous thing. Okay, so with the major diseases like anthrax, plague, they will not do this. Okay, this is a simple guess that you can make in the examination, but don't go with it. If you are sure, like if you can eliminate other options with a 40-60% chances, then go for this method of elimination. Otherwise, don't take a risk, because there is negative marking also. Next, question number 65. Covaxin, India's indigenous COVID-19 vaccine, was developed by. The right answer for this will be Bharat Biotech. Next, question number 66. The byproduct of a bioluminescence reaction is. So what is the byproduct of a bioluminescence reaction? The right answer for this will be option B, that is AMP. Remember what happened? The luciferin, that is the bioluminescent agent, plus ATP. Okay, ATP again plus oxygen, it give rise to oxyluciferins. Okay, oxyluciferin plus AMP plus light. Okay, so the right answer is AMP. AMP is released, so option B will be the right answer. So for those who don't know, let me tell you, luciferin. Okay, luciferin. Okay, luciferin is what? Luciferin is an organic molecule that is found in bioluminescent organisms. This undergoes oxidation and then light is emitted. Question number 67. An example of passively operated fishing gear is, okay, let me tell you, there are different types of fishing gear. The two major type of fishing gears is active and a passive gear. What is active gear? Active gear is something, active type of fishing nets are something in which the fish are being dragged. Okay, they are being dragged in the net. And passive type of fishing gear are something in which the fish swims, and in the course of swimming it gets trapped into the nets. Okay, so these are the two types of fishing gears present. So the right answer for this will be option C, that is the gill net. So this is example of a gill net in which the fish is passively swimming and getting trapped into the nets. Okay, next question, question number 68. Question number 68 is, the saucer shaped craft used in reservoirs for fishing in South India is. Okay, most of the people don't know. Let me show you the diagram of each and every type of fishing craft. Option A, which is the catamaran, it looks like this. Option B is coracle. This is looking like this. Then option C is your machua. Machua looks like this. And option D is your raft. Raft looks like this. So now you can make a good guess which one must be used by the South Indian people. It is option B, that is the coracle. Okay, coracle is used. Option C, that is the machwa, is used traditionally by the Maharashtrian. Okay, it is a Maharashtrian fishing boat. Next, moving on to next question number 69. Question number 69 is Forest Conservation Act in India came into force from the year. Option A is 1972. Option B is 1978, option C is 1980, option D is 1982. So Forest Conservation Act came in the year 1980. That is the option C will be the right answer. Okay, what happened in 1972? 1972 is the Wildlife Protection Act. Okay, Wildlife Protection Act. Then 1980 is the Forest Conservation Act. And there is no such acts present in 1978, 1982 as such. Okay. Next, moving on to question number 70. A global agreement to protect the stratospheric ozone layer was passed in Kyoto Protocol, Rio Conference, Montreal Protocol or Stockholm Convention. So the right answer for this will be Montreal Protocol. Okay, option C. What happened in other protocols? Let me tell you. Kyoto Protocol was for greenhouse gas. Okay, greenhouse gases. Then your Rio Conference. Rio Conference was a Earth Summit. Okay, it was the Earth Summit. Then your Montreal Protocol was for ozone. So the right answer for this will be Montreal Protocol. Next, question number 71. The species which are confined to a particular region and not found anywhere else are called pandemic. No, it is B, that is endemic. So this is the definition of endemic itself. There is nothing more to explain here. Then question number 72. The scientific name of the moth which produces tussar is Bombic mori. No, this is the mulberry silk. Anthria assamis. No, this is also wrong. Then this is Anthria assamis is for Muga silk. Okay, this is for Muga silk. Then again your Philosomia ricini. This is for your eri silk. And Anthria mylitta, this is for your tussar silk. So option D will be the right answer. Next question, question number 73. Local swelling of muscles and necrosis of kidney of fish is a symptom of. Okay, the right answer for this will be option D, that is Aeromonas infection. So option D is right. What are other? Let me tell you. Furunculosis. Furunculosis is boil-like lesions are present in the fishes. Second one is vibrosis. Vibrosis is a hemorrhagic disease. Okay, this is a hemorrhagic disease. So third option is dropsy. Dropsy means there is fluid accumulation. It is asking about local swelling. The local swelling and necrosis in kidney is option D. Next, question number 74. Question number 74 is, which of the following is regarded as the best egg producing chicken? So the best egg producing chicken is option A, that is Leghorn. Okay, option B, that is Rhode Island Red. This is used for dual purpose, both for egg as well as meat. Then option C, that is the Plymouth Rock. This is also used for dual purpose, that is meat as well as egg. And Ostra White is used for moderate. Next, question number 75. Which of the following fever is not transmitted by louse? The right answer for this will be Meiosis. Okay, option C is the right answer. Next, question number 76. The first recombinant DNA molecule was synthesized by Paul Berg et al. In the year. Which year? First recombinant DNA it is asking. This is a question from biotechnology and the answer for this will be option number B, that is 1972. Next, question number 77. Which of the following is true about restriction endonucleases? Let's read the options. Option A is type 1 and type 2 requires ATP to move along DNA. So this is wrong. Type 1, type 2 does not need ATP. Okay, so this is wrong because it is mentioning that type 2 also requires ATP. This is wrong. Then option B is type 1, type 2, type 3 requires ATP to move along. No, this is wrong because type 2 does not require ATP. Next, option C, Type 2 requires no ATP and cleaves DNA within recognition sequence. So this is correct because type 2 requires no ATP and it cleaves the DNA within the recognition sequence. So right answer will be option C. 78. Which of the following sponge is harmful for pearl oyster? So this is which sponge is harmful? The boring sponge is harmful. The boring sponge, and the boring other name of boring sponge is your Cleona. Cleona is otherwise called boring sponge, and this is harmful for ship industry, the shipping industry, for the pearl oysters, oyster farming who are doing for the pearl purpose. If Cleona, this sponge is present, boring sponge is present, it will harm the industry enormously. Next, 79. Which of the following statement is not correct? London smog is formed in winter. What is smog basically? Your fog plus smoke together makes what? Smog. They form smog. So what is smog? Smog is formed during winter. So this is correct. London smog is formed in winter. This is correct. Los Angeles smog is photochemical in nature. Okay, Los Angeles smog is photochemical in nature. This is also correct. London smog is reducing in nature. Yes, it is reducing. And London smog is sulfurous smog. Yes, it contains high amount of sulfur dioxide. So it is sulfurous in nature and it is generated due to coal burning. That is why it is reducing in nature as well. So all the A, C, D, all are actually correct options. This is a very tricky question. All the options are correct. So I think you can mark any of it according to your convention. For me, since Los Angeles smog is a different option other than London smog, if I would have been in your place, I would have marked B. But all the options are correct in this question. Next, question number 80. Question number 80 is HIV is, okay, HIV is which type of virus? This is asking. The right answer for this will be option A, that is the lentivirus. HIV belongs to lentivirus category. Adenovirus category are respiratory virus. Gellivirus is not the right options. Then papillomavirus, this is also irrelevant because papillomavirus causes warts. Okay, this cause what? So the right answer for this will be option A, that is lentivirus. So this is all for your CPET 2024 question discussion. I think you are working hard. If you are working hard, then remember, hard work never goes in vain. And please, students, do practice the questions of the previous year because I see most of the questions are being repeated. Like your gene therapy question is getting repeated. Silkworm question is getting repeated. So all the best, students, for your upcoming examinations. So that's all for today's video. See you in the next video.